Okay students, so the next figure, that is the transcription unit. Remember studying transcription? Yes? Okay. I don't want anybody talking. So transcription. Before going to transcription, let us see what is central dogma. What is central dogma? Right, okay. Central dogmavanna propose ಮಾಡಿದವರು ಫ್ರಾನ್ಸಿಸ್ ಎಚ್ ಸಿ ಕ್ರಿಕ್. ಇದನ್ನ ಪ್ರಪೋಸ್ ಮಾಡಿದವರು ಯಾರು? ಎಸ್, ಫ್ರಾನ್ಸಿಸ್ ಎಚ್ ಸಿ ಕ್ರಿಕ್ ಅವರು. Now central dogma, this gives us the flow of genetic message from DNA to RNA, DNA to RNA, and from RNA to protein. Yes, DNA to RNA is transcription. RNA to protein is translation. Right? Now DNA synthesizing DNA. What is this? Replication. Is it not? DNA synthesizing DNA, making an exact copy of itself, is known as replication. Yes. Now the central dogma got modified. Central dogma got modified after the discovery of an enzyme. Central dogma untalla idu what happened? Modify aitu after the discovery of an enzyme. Now this enzyme is reverse transcriptase. It got modified after the discovery of an enzyme, that is reverse transcriptase. Now this enzyme here, this enzyme helps in the synthesis of DNA from RNA, and this is seen in case of retroviruses. AIDS virus is a retrovirus. The genetic material of retrovirus is RNA. Now here RNA synthesizes DNA, right? And this process is known as reverse transcription, or it is also known as feminism, right? So ee process anna reverse transcription athava feminism anta helodu. So idanna discover madidavaru Howard Temin matte David Baltimore annuva scientist. So this feminism anna discover madidavaru Howard Temin and David Baltimore. That is why it is known as feminism, right? Okay. Now DNA synthesizing RNA is known as transcription, right? Okay. Iga ee transcription alli that you have studied the transcription unit, is it not? So we know that one of the strand of DNA runs in the direction 5 prime 3 prime, right? And the other strand 5 prime 3 prime, right? So out of the two strand, only one strand is involved in the synthesis of RNA. ಒಂದು ಸ್ಟ್ರಾಂಡ್ ಮಾತ್ರ ಇನ್ವಾಲ್ವ್ ಆಗಿರ್ತದೆ ಅಲ್ವಾ? the strand of DNA that is involved in the synthesis of RNA, ಈ ಸ್ಟ್ರಾಂಡ್ ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್. The strand that is involved in the synthesis of RNA. ಈ ಸ್ಟ್ರಾಂಡ್ ಅನ್ನ ಟೆಂಪ್ಲೇಟ್ ಸ್ಟ್ರಾಂಡ್ ಅಥವಾ ಈ ಸ್ಟ್ರಾಂಡ್ ಅನ್ನ ಆಂಟಿಸೆನ್ಸ್ ಸ್ಟ್ರಾಂಡ್ ಅಂತ ಕೂಡ ಹೇಳ್ಬಹುದು. Strand is also known as antisense strand, right? And the other strand, opposite strand, which is not involved, this strand is known as the coding strand or the sense strand. This strand is right, the coding strand, right? Yes. Now, transcription unit, the first gene of the transcription unit is promoter region ಅಥವಾ promoter gene. So, promoter gene is present at the 3' end of the template strand and 5' end of coding strand, right? Now the promoter gene in prokaryotes ಅಲ್ಲಿ ಈ promoter gene ಉಂಟಲ್ಲ, this is made up of sequence TATAAT, thymine, adenine, thymine, adenine, adenine, thymine, and this is known as Hogney's box. Prokaryotes ಅಲ್ಲಿ ಈ region ಅಲ್ಲಿ ಎಂತ ಇರೋದು? TAT. ಇದನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಪ್ರಿಬ್ನೋ ಬಾಕ್ಸ್. ಪ್ರಮೋಟರ್ ರೀಜನ್ ಅನ್ನ ಪ್ರಿಬ್ನೋ ಬಾಕ್ಸ್ ಅಂತ ಹೇಳೋದು, ರೈಟ್? ದಟ್ ಈಸ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಪ್ರೊಕ್ಯಾರಿಯೋಟ್ಸ್. ಅದೇ ಯೂಕ್ಯಾರಿಯೋಟ್ಸ್ ಅಲ್ಲಿ, ಇನ್ ಕೇಸ್ ಆಫ್ ಯೂಕ್ಯಾರಿಯೋಟ್ಸ್, promoter gene is made up of thymine, adenine, thymine, adenine, right? And this is known as Hogney's box. So in prokaryotes ಅಲ್ಲಿ promoter gene ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? ಪ್ರಿಬ್ನೋ ಬಾಕ್ಸ್ ಅಂತ ಹೇಳೋದು. ಅದೇ ಯೂಕ್ಯಾರಿಯೋಟ್ಸ್ ಅಲ್ಲಿ it is known as Hogney's box. The next region, this is the structural gene, right? Structural gene. And the last region here, yes, terminator. Terminator gene, right? Yes. Now, when is transcription taking place? G1 ಮತ್ತೆ G2 phase ಅಲ್ಲಿ. S phase ಅಲ್ಲಿ replication ಆಗೋದು, G1 G2 phase ಅಲ್ಲಿ transcription occurs, right? Synthesis of different types of RNA takes place, right? So what happens here? First, transcription ಅಲ್ಲಿ first step ಏನು? Initiation ಅಲ್ವಾ? First step is right, initiation. So what happens during initiation, students? An enzyme, that is RNA polymerase. This enzyme is also known as DNA dependent RNA polymerase. So the RNA polymerase enzyme comes and binds to which region? Promoter gene of which strand? Template strand. ಯಾವ enzyme bind ಆಗೋದು? Yes, RNA polymerase goes and binds to the promoter gene. So for this binding, one factor is required. ಒಂದು factor ಬೇಕು ಇದಕ್ಕೆ. Now that factor is called the sigma factor, right? So sigma factor is required for initiation. Right? Yes. So the synthesis of RNA begins. Opposite to which strand? Template strand. So if this is 5 prime end, here the RNA that is synthesized, it has got 3 prime end. So ಈ RNA polymerase enzyme promoter gene ಗೆ bind ಆಗ್ತದೆ, sigma factor help ಮಾಡ್ತದೆ ಈ binding ಗೆ, ಆನಂತರ sigma factor drops down, over, its function is done, it drops down, right? Next, second step is, yes, elongation. Elongation. So what happens during elongation? RNA polymerase enzyme moves on the template strand. Yava enzyme? RNA polymerase enzyme, the same enzyme moves on the template strand, and as it moves, what happens? RNA strand is getting synthesized. RNA strand synthesize ಆಗ್ತಾ ಹೋಗ್ತದೆ. Right? So that is elongation. The last step, third step, termination. So what happens during termination? I said RNA polymerase enzyme moves on the template strand, right? And synthesis of RNA is getting continued, right? Yes. Finally, the RNA polymerase enzyme reaches the terminator gene. ಎಲ್ಲಿಗೆ ರೀಚ್ ಆಗ್ತದೆ RNA polymerase enzyme? Terminator gene ಹತ್ರ reach ಆಗ್ತದೆ, right? And at that time one factor binds to it. This factor is known as the rho factor. Initiation ಅಲ್ಲಿ ಯಾವ factor help ಮಾಡೋದು? Sigma factor. Termination ಅಲ್ಲಿ ಯಾವ factor help ಮಾಡೋದು? Rho factor. Initiation ಅಲ್ಲಿ ಯಾವ enzyme helps? RNA polymerase. Elongation ಅಲ್ಲಿ ಕೂಡ RNA polymerase. Termination ಅಲ್ಲಿ ಕೂಡ RNA polymerase. Right? Yes. Now when rho factor binds to the RNA polymerase enzyme, what happens? Synthesis of RNA stops. RNA synthesis stop ಆಗ್ತದೆ. Right? Now the RNA strand gets detached from the template strand. Template strand ಇಂದ RNA strand detach ಆಗ್ತದೆ. ಅಲ್ವಾ? 5 prime end, 3 prime end. Yes. Now this RNA in prokaryotes is a functional RNA. Prokaryotes alli the RNA that is synthesized, what is it? Functional RNA. Functional RNA. Ade nammalli eukaryotes alli the RNA that is synthesized is not functional. ಇದು functional ಅಲ್ಲ. ಈ RNA ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? HNRNA, heterogeneous nuclear RNA ಅಂತ ಹೇಳೋದು. ನಮ್ಮಲ್ಲಿ ಈ RNA ಅನ್ನ HNRNA ಅಂತ ಹೇಳೋದು. That is heterogeneous nuclear RNA. Yes. Right? Now this RNA is not functional, students. It is not functional. Now for it to become functional, it has to undergo certain modification. ಇದು functional ಆಗಬೇಕು ಅಂತ ಇದ್ರೆ it has to undergo certain modifications. Now what are these modifications? ಈ modification ಅನ್ನ post transcriptional modifications ಅಂತ ಹೇಳೋದು. ಈ modifications ಅನ್ನ ಎಂತ ಅಂತ ಹೇಳೋದು? Post transcriptional modification. Now the RNA is synthesized. This is the 5 prime end. This is the 3 prime end. What type of RNA is this? hnRNA, non-functional RNA. Now what are the post transcriptional modification? There are 3 types of post transcriptional modification. One is capping. First is capping. So addition of cap region to the 5 prime end of hnRNA. Addition of what? Cap. What is cap made up of? Cap is made up of methylated guanosyl triphosphate. Cap ಅಲ್ಲಿ ಎಂತ ಇರೋದು? Methylated guanosyl triphosphate. Methylated guanosyl triphosphate. So post transcriptional modification ali first step is addition of cap region. Cap region to which end of hnRNA? 5 prime end. What is the cap region made up of? Methylated guanosyl triphosphate. Yes. The second step is splicing. Second step, splicing. Now what is splicing? Yes, ಇಲ್ಲಿ ಎರಡು regions ಇದೆ. ಈ hnRNA ಅಲ್ಲಿ there are 2 regions. One is the intron, another is the exon. So we have 2 regions, intron, exon. So there is intron, exon. Again there is intron, exon. Intron is the non-coding region. Intron is the non-coding region. Exon is the coding region. Now what is needed? Exon is needed. Intron is not needed. So splicing andre removal of introns and joining, not addition, joining of exons. And when you remove the introns, what happens? The size of hnRNA reduces. It becomes small, right? So this, splicing removes intron. So what is happening? 5 prime end alli what is present now? Cap is present. Introns are removed. Exons are joined. So this is splicing. Spliceosome ಅನ್ನುವ complex helps in this process. Spliceosome. Right? Yes. The third step, this is the second step. Third step is tailing. Now what is tailing? Tailing is the process of addition of poly A tail. To which end? 3 prime end of hnRNA. Addition of what? Poly A tail. Now what is this poly A tail made up of? What is it made up of? 20 to 200 adenine nucleotide. Idರಲ್ಲಿ ಎಂತ ಇರೋದು? Yes, adenine nucleotide. Now all these processes in eukaryotes takes place in the nucleus, right? And after that the RNA becomes functional. Now this RNA is the functional RNA, and this functional RNA comes out of the nucleus through nuclear pore. ಈ functional RNA nucleus ಇಂದ ಹೊರಗಡೆ ಬರ್ತದೆ, right? Through which one? Nuclear pore. And then this RNA will go to which one? It comes to the cytoplasm. After that this RNA goes to the ribosome. For what? protein synthesis. Right? DNA cannot come out. DNA ಗೆ ನ್ಯೂಕ್ಲಿಯಸ್ ಇಂದ ಹೊರಗಡೆ ಬರಲಿಕ್ಕೆ ಆಗುದಿಲ್ಲ. ಅದಕ್ಕೆ DNA ಎಂತದನ್ನ ಮಾಡ್ತದೆ? It cannot come out of the nucleus. So what is it doing? It is synthesizing RNA. Right? And this RNA comes out of the nucleus. It is taking the message from the DNA, genetic message and DNA in the tekoltade. Anantara comes out of the nucleus to the cytoplasm, and then it goes to the ribosome for what? Protein. Yes, to synthesize protein. Okay, so let's look into our question. Given diagram represents the components of a transcription unit. Select the correct answer regarding it. Transcription unit is on the board. Can you identify the right option? What is A, B, C and D? Option? Option C. Right. Yes. A is promoter, B is terminator, C is template, D is coding. Right. Okay. Next question. Match column 1 with column 2. Operator site, promoter site, regulator gene, structural gene. Yes. Match it. Idi yavudradu operator site, promoter site, regulator gene, structural gene. Yes, lac operon concept idu. Lac operon concept, this was studied in E. coli. Lac operon concept was studied in E. coli, right? Yes. Now, lac operon concept alli, this is the DNA, correct? DNA, right? DNA alli, which are the different genes present? Which are the different genes present? Yes, can you all tell me here? Which are the genes present here? DNA alli? Structural gene, right? Structural gene, structural gene alli ಏನೆಲ್ಲ ಇದೆ? lac Z, lac Y, lac A. Correct? Lac Z, lac Y, lac A. Lac Z codes for which protein? Beta galactosidase. Lac Y codes for lactose permease. Right. Lac A codes for trans acetylase. DNA synthesizes RNA. DNA is synthesizing RNA, and RNA is synthesizing these proteins when it goes to the ribosome. Correct? So, DNA synthesizing RNA is transcription, RNA synthesizing protein is translation. Right? Yes. The next region, which is the next region? Operator gene. Operator gene, represented as lac O. The next region, promoter, represented as lac P. And promoter gene is the binding site for which enzyme? RNA. Promoter region ಗೆ ಏನು ಬಂದು ಬೈಂಡ್ ಆಗೋದು? RNA polymerase. Correcta? Last region, regulator gene, lac I. ಏನನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತದೆ? DNA is producing RNA, RNA is producing repressor protein. And repressor protein goes and binds to operator. Right? Repressor protein ಯಾವುದಕ್ಕೆ ಹೋಗಿ ಬೈಂಡ್ ಆಗ್ತದೆ? ಆಪರೇಟರಿಗೆ ಹೋಗಿ ಬೈಂಡ್ ಆಗ್ತದೆ. When lactose is not present. And when lactose is present, repressor protein binds to lactose. In the absence of lactose, lactose ಇಲ್ಲದಾಗ repressor protein ಎಲ್ಲಿಗೆ ಹೋಗಿ ಬೈಂಡ್ ಆಗ್ತದೆ? ಆಪರೇಟರಿಗೆ ಹೋಗಿ ಬೈಂಡ್ ಆಗ್ತದೆ. When lactose is present, repressor protein goes and binds to lactose. Right? Yes. Okay, now can you tell me the right option? Match it and tell me. Yes? Option B, right. Second option. Now just now I showed you the mRNA, is it not? Post-transcriptional modifications alla maadi we have seen the mRNA. So can you tell me what is A, B, C and D? Their A is methylated group, methylated cap. B is non-coding region. C is the coding region. And D is poly A tail. No, I have been given this paper. What's the 12th question that you all have?
Which of the following function of RNA?
Which of the following?
Function of
Which of the following is a function? So many questions are not there. Histone protein idea. How many questions have you all finished now? 10th. 11th? You don't have that question? Your 11th question is something different. Okay, my 11th question is something different. Okay, so what's your 11th question? Histone proteins are, what are they? Basic, positively charged. Yes, it is basic and it is positively charged. Yes. Which of the following is a function of RNA? RNA, it is a carrier of genetic information from DNA to ribosome, synthesizing polypeptides. Is it right? RNA ಏನು ಮಾಡ್ತದೆ? DNA ಇಂದ ಇನ್ಫಾರ್ಮೇಷನ್ ಅನ್ನ ತೆಕೊಳ್ತದೆ, ಆನಂತರ ರೈಬೋಸೋಮಿಗೆ ಹೋಗ್ತದೆ for synthesizing proteins ಅಥವಾ polypeptides. Right. It carries amino acid. tRNA ಏನು ಮಾಡ್ತದೆ? ಅಮೈನೋ ಆಸಿಡ್ ಅನ್ನ ಕ್ಯಾರಿ ಮಾಡ್ತದೆ. ಎಲ್ಲಿಂದ? From the cytoplasm to the ribosome. For what? For protein synthesis. Right? Yes, that is also right. It is a constituent component of ribosome. rRNA. rRNA is the constituent of ribosome. rRNA is what? Ribosome. ಮೂರು ತರದ RNA ಗೊತ್ತಿದೆಯಲ್ಲ? mRNA, tRNA, rRNA, right? mRNA, messenger RNA. What is the concentration of messenger RNA in the cytoplasm? ಕಾನ್ಸಂಟ್ರೇಷನ್ ಎಷ್ಟು ಮೆಸೆಂಜರ್ ಆರ್ ಎನ್ ಎದು ಸೈಟೋಪ್ಲಾಸಮ್ ಅಲ್ಲಿ? Yes, its concentration in the cytoplasm is somewhere around 2 to 5%, right? Messenger RNA the concentration irodu cytoplasm alli 2 to 5%. Yes, messenger RNA is a linear RNA. It is a single stranded RNA, linear RNA, straight RNA, right? And messenger RNA is a short-lived RNA. It lives for a very short duration. So it is a short-lived RNA. Messenger RNA untalla, this is a short-lived RNA, right? Yes. Messenger RNA are of two types, polycistronic, monocistronic. ಎರಡ್ ತರದ್ದು, polycistronic ಮತ್ತೆ ಮೊನೊಸಿಸ್ಟ್ರಾನಿಕ್. ಪಾಲಿಸಿಸ್ಟ್ರಾನಿಕ್ mRNA ಯಾವುದ್ರಲ್ಲಿ ಇರೋದು? ಪಾಲಿಸಿಸ್ಟ್ರಾನಿಕ್ mRNA, this is present in prokaryotes. This is present in prokaryotes. ಯಾವುದು? Polycistronic mRNA. And polycistronic mRNA codes for several proteins at a time. It can produce several proteins. Poly alva, it can code for several proteins, right? It can code for several proteins at a time. The monocistronic mRNA is present in, yes, eukaryotes, and this can code for only one protein at a time. So it codes for only one protein. Yaudu? Monocistronic mRNA, right? Yes. Second one is, which is the second type of RNA? tRNA or transfer RNA. tRNA ಅಥವಾ transfer RNA. Yes. Now tRNA or transfer RNA, this is the shortest RNA. Short-lived ಯಾವುದು? mRNA. Shortest RNA ಯಾವುದು? tRNA, and its concentration in the cytoplasm is around 15%. Right? Now the structure for tRNA was given by Robert Hawley. Structure for tRNA was given by who? Robert Hawley. Have you all studied this structure, students? Is structure odidira? What is this structure called? ಓದಿದಿರಿ ಅಂತ ಹೇಳ್ತೀರಾ. What is the structure called? This structure is known as the clover leaf model. ಈ ಸ್ಟ್ರಕ್ಚರ್ ಅನ್ನ ಕ್ಲೋವರ್ ಲೀಫ್ ಮಾಡೆಲ್ ಅಂತ ಹೇಳೋದು, right? This resembles the trifoliate leaf of clover plant. ಇದು ಕ್ಲೋವರ್ ಪ್ಲಾಂಟ್, ಇದು ಟ್ರೈಫಾಲಿಯೇಟ್ ಲೀಫ್ ಅನ್ನ ರಿಸೆಂಬಲ್ ಮಾಡ್ತದೆ. ಸೊ ದಿಸ್ ರಿಸೆಂಬಲ್ಸ್ ದ ಟ್ರೈಫಾಲಿಯೇಟ್ ಲೀಫ್ ಆಫ್ clover plant. That is why it is known as the clover leaf model, right? So can you see these arms? What are these arms? In arms ivugalu, this is the first arm, right? What is this arm called? Amino acid ಅಥವಾ acceptor arm ಅಂತ ಹೇಳೋದು. This arm is known as acceptor arm, right? Acceptor arm. function ಎಂತದು? Its function is to bind to the amino acid. ಯಾವ ಎಂಡಿಗೆ ಬೈಂಡ್ ಆಗೋದು? ಅಮೈನೋ ಆಸಿಡ್ 3' ಎಂಡಿಗೆ ಬೈಂಡ್ ಆಗೋದು. ಸೊ ದಿಸ್ ಈಸ್ ಅಮೈನೋ ಆಸಿಡ್ ಬೈಂಡಿಂಗ್ ಸೈಟ್. Amino acid binding site, right? And here the nitrogenous base that is present is CCA, cytosine adenine. So what is the first arm? Acceptor arm. Acceptor arm alli you can see this has got a stem. Adre ee armige loop is not there. Loop illa, right? The next arm, what is this arm? This arm is known as T-Zy-C arm ಅಥವಾ pseudo uridine arm. T-Zy-C arm ಅಥವಾ pseudo uridine arm, right? Now this arm you can see it has got a stem and it also has got a loop, correct? And this arm binds to the ribosome during protein synthesis. ಈ ಆರ್ಮ್ ಯಾವುದಕ್ಕೆ ಬೈಂಡ್ ಆಗ್ತದೆ? ರೈಟ್, ಟು ದ ರೈಬೋಸೋಮ್. ಯಾವಾಗ? ಪ್ರೋಟೀನ್ ಸಿಂಥಸೈಸ್ ಆಗುವಾಗ, ರೈಟ್? ನೌ ದ ನೆಕ್ಸ್ಟ್ ಆರ್ಮ್, ದಿಸ್ ಥರ್ಡ್ ಆರ್ಮ್ ಯು ಕ್ಯಾನ್ ಸಿ, ಈ ಆರ್ಮ್ ಅನ್ನ ವೇರಿಯೇಬಲ್ ಆರ್ಮ್ ಅಂತ ಹೇಳೋದು. The third arm is known as the variable arm. Now variable arm untalla, this is not present in all tRNAs. This is present only in few tRNA and the function of the variable arm is still unknown. Idra function innu kuda discover agilla, right? Yes. The next arm is, what is this arm? Anticodon arm. The next arm is known as anticodon arm. Now if you see the anticodon arm, students, anticodon arm has got a stem and it has got a loop. Stem is there, loop is also there, right? Now this anticodon arm, what is the function? Idradu loop untalla, and the loop has got 3 nucleotides. Enide illi 3 nucleotides galu untu. And the function of this arm is to bind to the mRNA. Yavudakke idu bind agtade? Yes, it binds to the mRNA. Yavaga? Protein synthesis aguvaga, right? It binds to the mRNA, right? mRNA alli enta irodu? Codons. mRNA contains codons. tRNA alli enirodu? Anticodons, right? ಈ ಕೋಡಾನ್ ಅನ್ನ ನೀವು ಅಪೋಸಿಟ್ ಡೈರೆಕ್ಷನ್ ಅಲ್ಲಿ ಓದಿದ್ರೆ this is called no doc. No doc. ಅದಕ್ಕೆ codon is also known as anticodon is also known as, ಈ ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ಸ್ ಅನ್ನ ಏನಂತ ಹೇಳೋದು? no doc ಅಂತ ಹೇಳೋದು, right? Anticodon is also known as no doc, right? Anticodons are present on tRNA. Codons are present on mRNA. Anticodons are also known as no doc, right? Yes. The last arm, what is this arm? Yes, DHU arm, the last arm, right? DHU arm. DHU stands for dihydrouridine arm. DHU andre dihydrouridine arm, right? Now this arm, what is the function of this arm? arm binds to one enzyme and that enzyme is amino acyl synthetase enzyme. So ಈ ಆರ್ಮ್ ಯಾವುದಕ್ಕೆ ಬೈಂಡ್ ಆಗ್ತದೆ? amino acyl synthetase ಅನ್ನುವ ಎಂಜೈಮಿಗೆ ಬೈಂಡ್ ಆಗ್ತದೆ. ರೈಟ್?